Hello, my name is Belinda Uwase, and I'll be going over the gut microbiome experiment. A microbiome is a collection of microorganisms. The gut microbiome specifically consists of trillions of microorganisms that live in the intestinal tract. This helps with food digestion, immune system regulation, and protecting us from pathogenic invasions. From a physiological standpoint, studies show that there's been correlation between the gut microbiome and human metabolism, nutrition, and disease. Having an imbalance of the microbiome can lead to many gastrointestinal health problems, such as type 2 diabetes, obesity, and inflammatory bowel disease. Although type 2 diabetes can be hereditary, you can get it from the compost of the microbiota. Having an incline of butyrate producing bacteria that regulates energy metabolism. Our microbiome has developed over time to adapt to our environment and diet. The microbiome does not only affect our immune system, but also the gut connected to the brain connection. The endocrine and immune system have direct communications with the brain due to self-produced neurotransmitters and nerve receptors in the gut. This maintains optimal levels of vitamins and minerals and healthier diet, which makes for better brain functions. For our hypothesis, we asked, as the weight of the German Shepherd increases, the function of their microbiome decreases compared to those who weigh less, making it more likely for them to develop gastrointestinal diseases. The German Shepherds we use all ranged from a various of weights. 365 weighed 80 pounds, was 10 months old, and was a male. 406 is 70 pounds, 1 year old, 5 months, and is a female. 521 was 67 pounds, 1 year and 4 months old, and was also a male. 643 was 80 pounds, 2 years and 5 months old, and was also a male. Here's a quick overview of our experiment. We isolated and concentrated the DNA, isolated the 16S rRNA gene, then took readings from gel electrophoresis, went through applicon cleanup, and indexing PCR. After all of those steps, we then cleaned up the PCR and put our samples in a fluorometer after to get an accurate reading of the bacteria that we found in our DNA. We first isolated the DNA from sterile fecal samples using the QIAGEN kit, which is designed for DNA isolation. After that, we concentrated the DNA sample by using the Zymo kit. The isolation is based on blending the stool in the buffer, then that prevents enzymes from degrading the DNA. After that, we then binded the DNA to a filter embedded in a small column using a series of rinses to wash away impurities, and finally unbind the DNA from the matter in a buffer that weakens the DNA and the filter bond. PCR amplification. We want to isolate the 16S rRNA gene because it consists of 9 variable regions. After that, those regions are sequenced and will tell us what type of bacteria is present in the gut. We are then going to use gel electrophoresis to remove primers, DNTP, and other PCR reagents using a PCR cleanup kit. After that, we will add sequence tags to the end of the applicon from each sample in an indexing PCR reaction. The tube also went through a thermal cycle of 95 degrees for 30 seconds, 55 degrees for 30 seconds, and 72 degrees for 30 seconds. This went through 8 cycles, as where applicon PCR was 25 cycles. After all of that, we're then going to rinse off the impurities and then elute in another buffer. This makes the DNA 10 times more concentrated. As you can see by the picture, we have a representation of our gel electrophoresis results. As you can see, our positive control are German Shepherd 643, 406, and 1040 showed up in our reading. Then we have to clean up the PCR reaction products using the same kit quantifying the amplified DNA to get its reading from the sequencing. The cleanup will remove the compost of the amplified PCR buffer as well as the unused primer. Now we're going to use a fluorometer. We'd rather use a fluorometer than a spectrophotometer for a more accurate reading. The picture tables for week 2 were created by the group in our section that shared the same DNA as us. Unfortunately, we were not able to provide pictures of our own calculations, but we did ask their permission for their calculations because we use the same samples. As you can see, the ratios all lie within 1.5 to 2.2, which would indicate where pure DNA would reside. The values below 1.5 indicate contamination with proteins. So we likely have pure DNA. We then wanted to create 50 microliters. Each solution contained 27 microliters of Maxter mix. We then used concentration of DNA in each sample to determine how much of the DNA template to add. The solution had brought up to 50 microliters by adding water. All of the samples had over 10,000 reads, all of which passed quantity filtering. This is the picture that we got from our neighboring group that shared the same samples as us. As you can see, it indicates our nano drop readings, also our PCR calculations. At the kingdom level, all of our samples were almost at 100% bacteria, which is ideal. At the order level, bacteriolase and castrolase were found in high levels in all samples except the positive control. At the genus level, Prevotella was the most common strand in all samples except for dog number 3, which was 521. Bacteriolase was the most common strand in dog 3 and the second most common in all samples except for dog 2, which is 406, which had high concentrations of Ferrobacterium. Once again, weight did not seem to correlate to concentrations of bacteria within the sample. This also indicates our bar graph for our genus level analyzation. For our unclassified results at the kingdom level, we had 5 for 365, we had 2 for 406, 7 for 521, 2 for 643, and 3 for our positive control. The most common readings in our order were bacteriolase, castrolase, fructose bacteriolase, Burkholderlase, and enterobacteriolase. The common genuses were Bacterioide, Prevotella, Baphytia, and Fusobacterium. We also had a little bit of Escherichia. In conclusion, the DNA extraction of scat was not successful due to the attentiveness of each set of procedures. We did not expect PCR product in our negative control, as we could see from our gel electrophoresis readings that there wasn't any indicated. And that will conclude this experiment. Thank you.